So in the last clip, the ending of my part 1, I was simply saying that we have 2 parts of our lab session, which are just emphasizing the diagnostic tools, and the part A, the microscopy, and the part B, the immunologic tests, with emphasis on an example on rapid diagnostic tests. These 2 parts, under each part, part A, for example, I have tasks set up for you, and when we mention tasks, it's basically the microscopy station, according to the diagnostic stage that you're required to observe. So there will be a task, okay, against that microscopy station. So I'll just run you through what that means, what the tasks are, of course, but these are our 2 parts of the session, part A and part B, emphasizing the 2 commonly used methods or tools. Yeah, diagnostic tools. Okay, so it's again this I'm just emphasizing in this slide. Part A is microscopy, and what this means is detection of the diagnostic stages. Okay, so diagnostic stages can be your eggs, your parasitic eggs, your larvae, your oocysts, your cysts, your trophozoites, your trypanosomes, whatever it is that is connected to the suspected parasite. Okay, that can be detected in the clinical specimens. And there are various options for the specimens, okay? Of course, according to the parasite of interest. But in the slide here, we can see we can have, for example, the fecal sample, okay? You can have blood as a sample, sputum, aspirates, CSF, and so on, okay? Any possible body fluid or scraping, anything where we can obtain from the host without harming the host, of course, and then also where we can look for certain morphological stages that are diagnostic. Okay, that's the scope of the microscopy. So we can just look at the tasks. What are the morphological stages that you will be appreciating in the session? So we have the very first task there, task 1, which is a trypomastigote of Trypanosoma bruceirodescens. Look at those very clear trypomastigotes. So you have this microscopic station showing exactly as shown here, and you're expected to appreciate this at least one of them. These trypomastigotes appreciate your kinetoplast, this very important structure, kinetoplast, okay, and the central nucleus, okay, and elongate structure, and so on, okay. Most important, at least I need to see these 2 features. And also comment on the GEMSA color. This is a GEMSA stained slide, so it's a very unique or rather important staining option in parasitology. Okay, so you have some nice red purple coming out in the nuclei material. Okay, so kinetoplast is mitochondrial DNA associated there, and then you have the main genetic material in the nucleus there. So there's a red purple because of GEMSA, so pay attention to that as well. So all these requirements or observations, the task, are going to be tabulated, okay, but this table you will fill in later on at your own time. The most important is a proper observation to generate a drawing. That is the focus as you come for that 1 hour. Just generate a very good drawing You don't even need to draw this table. I just need one single paper where you're giving some clear drawings of what you have observed, and then later on that signed sheet that will be attached to the report, later on, before you submit, of course, fill in according to this table. Okay, so this is your table 1 for that very first task. You have the specimen there. Of course, you have to comment in the first stage what is the specimen. In the first stage of trypanosomiasis, what is the specimen? And then, of course, it's clear there, morphological stages, trypomastigote, a labeled drawing, very clear labels, okay? Label all the parts that have been appreciated, classification, of course, and then comment, at least 2 comments relevant to the drawing, okay? And then final note on the tasks, for example, in this one, and all the tasks and all the tables that you're required to generate. The title should be very clear. This is not a complete title, dash, 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 right? I've shown you there. So please indicate correctly your titles, okay? Very important also. Indicate fully your title. Yeah. So I'll just go into the task 2 and quickly just explain this slide. You can see here these microscopic images. So this is what you will have on the bench. Of course, this is in the microscope in the demonstration slide, but also there is a bench aid, what we call a bench aid. Okay, and this bench aid it's almost like a confirmation image. As you look into the microscope, you see